Welcome back to the University of Melbourne Biology series. This is episode 11, and today we ask a question that sounds simple but reshaped the history of life. Why be more than one cell? A single cell trades materials directly with the water around it. Yet again and again, life gathered cells into larger bodies. That choice unlocked enormous possibilities, but it created a serious problem. How do you keep the cells buried deep inside a large body alive? Over the next 12 minutes we will trace how multicellularity arose, the benefits it brought, the physical limits it ran into, and the elegant solutions that life evolved to move gases in and out. Let us begin with how single cells first came together. Multicellularity is not a one-off invention. Remarkably, it arose at least 6 separate times in eukaryotic history, evolving again and again in different lineages. So how does a population of single cells become one organism? The transition likely unfolded through a series of steps. First, cells aggregate, gathering into a cluster instead of drifting apart. Next, those cells begin to communicate with one another. Then some of them specialize, taking on particular jobs rather than each doing everything. There is a cost to this. As cells commit to the group, they lose the ability to live independently. They become parts of a whole. The payoff for that sacrifice is increased size, functional specialization, and far more complex organisms than any single cell could be. What does a cell gain by giving up its independence? 2 big things. The first is size and complexity. A body of many cells can grow far larger than a lone cell and take on tasks no single cell could manage. The second is functional specialization, a division of labor in which different cells dedicate themselves to different jobs, making the whole organism more efficient and capable. And the results are everywhere. Multicellular life spans an astonishing diversity, from simple colonial organisms through to complex animals and plants. That diversity reflects the many independent evolutionary paths and adaptations multicellularity has taken. The same basic idea, cells living and working together, has been reinvented over and over into a dazzling variety of forms. But bigger is not free. Becoming multicellular carries real consequences that set the agenda for the rest of this episode. A large body needs complex transport systems to move materials around inside it. It carries increased metabolic requirements, demanding more energy than a simple cell. And because its cells are specialized and interdependent, it needs coordination and communication to keep them working together. These are not just inconveniences, they are genuine limitations on what a multicellular organism can be. Multicellular life only works because it found ways to meet these challenges through specialized systems and regulatory mechanisms. To understand why, we need to look at a simple piece of geometry. That piece of geometry is the surface area to volume ratio, and it quietly governs everything that follows. A cell exchanges materials across its surface, but it must supply its entire volume. Here is the catch. Small organisms have a large surface area to volume ratio, plenty of surface to serve a small interior. But as an organism grows larger, that ratio decreases, the volume races ahead while the surface lags behind. This ratio is a fundamental factor in multicellular life, because it determines the efficiency of diffusion. For a small body, diffusion alone can keep every cell supplied. For a large body, the shrinking ratio leads to real limitations in diffusion, and that is what forces large organisms to evolve specialized gas exchange structures rather than relying on their outer surface. So how do large organisms beat a problem that geometry seems to make unbeatable? They did not find one fix, they found 3, and they work together. The first innovation is to maintain an internal aqueous environment, a watery medium in which exchange can happen. The second is specialized transport systems, which make sure substances actually reach cells throughout the body. The third is specialized exchange surfaces, structures built to maximize gas exchange. Together these 3 adaptations allow efficient gas exchange and nutrient distribution throughout even a very large organism. Think of them as a team, an internal medium to exchange into, a network to carry materials around, and dedicated surfaces where gases cross in bulk. Let us take each one in turn, starting with the internal environment. Start with the first innovation, the internal environment. In a large organism, most cells never touch the outside world. Instead, they are bathed in an internal aqueous environment, and it is this medium that serves them as the place for gas exchange and nutrient distribution. Every buried cell can trade materials locally with the fluid around it, even if it sits far from the body surface. But there is a condition. That internal environment only works if it stays stable, and keeping it stable is the job of homeostasis. Homeostasis maintains the internal environment so that it remains stable and conducive to cellular function. So the first solution to being big is to carry a small, controlled, watery world inside you, one that every cell can rely on. An internal medium is only useful if materials can move through it, and that is the second innovation, specialized transport systems. Their job is to ensure that substances from specialized tissues and organs reach cells throughout the whole organism. In animals this is the circulatory system, in plants it is tissue such as the phloem. These systems do something subtle. By constantly carrying materials toward and away from cells, they maintain high concentration gradients for diffusion. Remember, diffusion needs a difference to drive it. A transport system keeps delivering fresh supply and sweeping away waste, so the gradient never runs flat. The result is efficient gas exchange and nutrient distribution across the body. The internal environment gives cells somewhere to exchange, the transport system makes sure the right materials keep arriving. Now the third innovation, the exchange surfaces themselves, and the law that explains their shape. Specialized exchange surfaces, such as the alveoli in the lungs and the spongy mesophyll cells in plants, share two features, a large surface area and a thin barrier. Why those two? The answer is Fick's law, which describes the rate of diffusion. Fick's law says the rate depends on four things: the surface area available, the partial pressure gradient across the barrier, the diffusion coefficient of the gas, and the diffusion distance it must cross. The rate rises when surface area or the gradient increases, and it falls as the diffusion distance grows. So a good exchange surface does exactly what the law rewards: it maximizes surface area and minimizes diffusion distance. One term in Fick's law deserves a closer look, because it is the force that drives a gas across a surface, the partial pressure gradient. In any mixture of gases, each gas exerts its own share of the pressure, called its partial pressure. The partial pressure of a gas is the pressure exerted by that one gas within the mixture. What matters for exchange is the difference in partial pressure from one side of a surface to the other. A gas diffuses from where its partial pressure is high to where it is low, so partial pressure gradients drive the diffusion process in gas exchange. Understanding partial pressure is therefore essential to explaining how gases move across any exchange surface, whether in a lung, a leaf, or a sponge. Let us see all of this come together in the clearest example, the human lung. The lung has a structure that maximizes gas exchange, optimizing every term in Fick's law at once. First, it has a very large surface area, which directly raises the rate of diffusion. Second, it has a thin barrier between air and blood, which minimizes the diffusion distance gases must cross. And third, it maintains a high partial pressure gradient between the air inside it and the blood passing through it, which keeps oxygen and carbon dioxide moving in opposite directions. Large area, short distance, steep gradient, these three properties facilitate efficient diffusion of oxygen and carbon dioxide across the alveoli and the capillaries. The lung is essentially Fick's law turned into an organ. The same rules govern organisms that look nothing like us. Take a plant. Plants exchange gases through stomata, small openings on the leaf surface. Carbon dioxide enters the leaf for photosynthesis, while the oxygen produced by photosynthesis diffuses out. Inside the leaf blade, the spongy mesophyll cells act as the specialized exchange surface. Now take a sponge, a simple aquatic animal. A sponge uses its body cavity and its collar cells for gas exchange. The flagella of those collar cells create a water current through the body, bringing in food and moving respiratory gases. And notice the pattern: the structure of the sponge and that water current are related to Fick's law, because together they maximize surface area and minimize diffusion distance, just as the lung and the leaf do. Step back and look at the whole arc of this episode. Multicellularity gave life size, specialization, and staggering diversity, but it came at a price written into simple geometry. As bodies grow, the surface area to volume ratio falls, and diffusion alone can no longer reach the cells inside. Life answered with three innovations working as one team. An internal aqueous environment, held steady by homeostasis, gives every cell a place to exchange. Specialized transport systems keep the right materials arriving and the gradient steep. And specialized exchange surfaces, obeying Fick's law with their large area, thin barriers, and strong partial pressure gradients, move gases in bulk. Lung, leaf, and sponge are three answers to the same question. From the origins of multicellularity to a single breath, it all connects. See you next time.